Welcome to a quick little introductory video / review video for your test on not only cellular respiration, but your test on photosynthesis. So I want to first start talking about the cellular respiration and what that all entails. We're going to go through and look at our equation for cellular respiration. We got to figure out what our products and our reactants are going to be. What do we need to actually get our end products? So why do you eat? Why do you breathe? So what we need to know is that we need to take in oxygen and we need to take in glucose in the equation C6H12O6, and we convert that into our end products, which is carbon dioxide and water. So if I'm going to zoom in on there a little bit, we want to make sure we have our equation balanced, and this will make more sense later on. 6 waters. This is our equation for cellular respiration. Now, the only reason you are able to take in oxygen, the reason you take in this food, this sugar, is again, only main reason is to make this guy, ATP. So how do we actually go about doing the creation of ATP? We got to first start off in the cell itself. We're looking at a cell, no matter if it's an animal, plant, bacteria, inside the cytosol. Cytosol or cytoplasm, we're going to undergo a process known as glycolysis. Glycolysis is going to take place, and what that really refers to is a fancy term of splitting that structure there, taking C6H12O6 and splitting that into two structures, C3, to get our C6s. Okay, we don't really need to get to too much detail. What's going to end up involved is that this C3 structure and that C3 structure is known as PGAL, phosphoglyceraldehyde. Okay, phosphoglyceraldehyde, PGAL is also known as G3. Now, zoom on to that. Now, in order to split that up into two different structures, I needed to expend some energy in the form of 2 ATP. So I had to use some energy up. But I want to take this G3P structure and I want to convert those into pyruvate. So your two molecules of pyruvate, and the pyruvate is again a structure, a 3-carbon structure. What I get out of each one is minus 2 ATPs here, I get 2 ATPs plus, and I get a plus NADH per pyruvate, so I multiply this times 2. Okay? So if we're looking at our end result, we're looking at our total end result of glycolysis. What are we going to go plus a net of 2 ATP and 2 Those are the things that are going to be taking place. Now again, this is happening in the cytosol and without oxygen. If I'm going to go through and I can talk about the process of still creating that energy, but in the cytosol, if there is no oxygen present, I go in a fermentation cycle. And that fermentation can be one of two things: lactate, more of a lactic acid fermentation, or number 2, ethanol. There are 2 different methods of fermentation in which I take this reduced form, NADH, and I convert it back to my oxidized form, NAD. And that allows for this electron carrier to get back to glycolysis and be ready if oxygen becomes present later on. The byproducts, though, are lactate in humans, lactic acid, and ethanol, things like in yeast or bacteria. Now, with oxygen being present, what we typically do see, we see with oxygen, we're going to get into this large organelle structure. or the inner mitochondrial folds, all known as the cristae. So inside here, that's where we're going to see the Krebs cycle, and in that inner membrane called the cristae, you are going to have the ETC. So let's kind of shrink this up a little bit and see, as we have our pyruvic acid, glycolysis is going to enter into the mitochondria. What's going to happen is I'm going to take this three carbon so okay. What happens to this loss? There goes the carbon dioxide. Now, the acetyl-CoA is going to get smaller. This guy enters into the matrix. So I have a 2-carbon of acetyl-CoA joins up with a 4-carbon structure, oxaloacetate, 6-carbon structure. As those form together, that 6-carbon structure is going to get rid and continue into a circle. 6, 5, 4, and I rearrange my 4 until I get back to the acetate. In the process, acetyl-CoA, acetyl-CoA, 1 ATP, 3 NADHs, and 1 NADH2. So reduced form. So I have a total of 5 electrons for acetyl-CoA and 1 ATP. Now, how many acetyl-CoAs are there? There are 2 of them. So where we could double all our things. We would have 2 ATP, 6 NADHs, and 2 FADH2s. Now, that being said, we're going to go from our mitochondria, those inner folds within here. Zoom into one of those folds, we just zoom in this box. What we're looking at is a phospholipid bilayer. Go through the entire detail of it, but inside there we have the ETC taking place. Now, what's going to happen is that we are going to be seeing those electron carriers. Let's look at that one right now. The electron of that is going to work its way through. It's been oxidized, so NAD becomes now, that should end in NADH becomes NAD. And that hydrogen ion works its way through. So I have all these H pluses on here, but the electron form of it bounces down throughout this electron transport chain. And at the very end of the electron transport chain, I have one last protein that's going to allow this electron to go through. And I have one final electron acceptor that is oxygen. And when that collects that last electron acceptor, what we're left with is molecular H2O. We get our water. Now, what we say with all those protons that are going to be pumped through, remember we've got a whole bunch of electrons that were being created, those reduced NADHs, as well as the FADH2s. We create this big gradient taking place throughout the membrane, and then we have this one proton or structure, protein structure, that's going to be embedded with the inside of this membrane. And as this hydrogen ions travel down through this, it's a big turning gradient. ATP plus inorganic phosphate, and I can stick them together. So what's turning out is ATP. Now, being that we used oxygen in this process, this is called oxidative phosphorylation. Okay? Now, what's it all entail is if we go through this process, glycolysis to ATP TCA to ATP, and then the electron up to 34 ATP. Now, we're not always going to get all of those energies from that. This is not going to get all of the energy source. We are going to get a lot of it. Ultimately, we uh, ATP molecules. So that is just a review process of cellular respiration. Very, very quickly, but you can go from there and select on the next video, which is just going to go quickly through the process of photosynthesis.